Thank you. Hi, everyone. My name is Amina. I'm a research technician at the Princess Margaret Genomics Center. I work on the spatial transcriptomics team, specifically on Visium HD and Visium V2. And today I'll be talking about the sample processing portion of Visium HD. I know that this is a bioinformatics conference, but I'm hoping that giving you a better understanding of the sample processing portion will give you a more holistic understanding of the Visium HD assay. Um, so this module is called garbage in, garbage out as referenced before. It's just referring to the quality of the data that you receive will be determined by the quality of the sample that you submit to us. And yeah, so feel free to ask any questions at any time. So this is just a quick overview of what I'm going to discuss. I'll start by discussing what is Visium HD and how does Visium HD work. And then I'll be going into the RNA quality control that you need to do before you submit Visium samples. After that, I'll be going in depth about the experimental protocol and what we do at the bench, and then the web summary that we receive and the key metrics that we look at before we send you the data. So what is Visium HD? So Visium HD is a spatial transcriptomics platform that was released by 10x Genomics. It allows for high resolution whole transcriptome gene expression analysis of tissue sections. And it's based off of previous Visium technologies. So I'm not sure if any of you have worked with the V2 version of Visium. It had two 6.5 by 6.5 millimeter capture areas on the slide and had about 5,000 barcoded spots that were 55 micron in diameter with some gaps in between. So HD is a higher resolution version of that. The same size capture areas, so 6.5 by 6.5 millimeter capture areas, two of them per slide. And they have 2 by 2 micron bins that span the entire capture area. So there's no gaps in between. And this allows for much higher resolution than we have for Visium for Visium V2. Similarly to Visium V2, it's pro-based. So that allows us to work with lower quality samples like formalin fixed paraffin embedded samples, FFPE samples, as well as fixed frozen samples. And we still can work with the higher quality fresh frozen samples as well. And it also allows a slight limitation to that is that we need to have probes for those samples, and 10x Genomics only has probes for human and mouse samples, so you're limited to those species as well. So how does Visium HD work? So it all starts with sample preparation. I'm not going to go too much in depth about sample prep, as Melody will be doing that later. But once we get the sections, we're ready to do deparaffinization and imaging. Deparaffinization is specific to FFPE samples. However, for all samples, we do image them. We either do an H&E stain or an IF stain. I'll be talking more about H&E staining as that's a lot more popular. However, you can do IF staining as well, immunofluorescence staining. Then we get to the processing in the cassette. So we have decross-linking and probe hybridization. Probe hybridization happens overnight. And then the next day we do the transcriptomic probe transfer, which happens in the actual cytosist. After that, we move on to probe extension and library construction. And once we have the libraries, they're ready to go for sequencing. We sequence the samples, and then you get the data. So what is RNA quality control? So we do RNA quality control before you do your Visium samples. Visium HD is quite an expensive assay, so it's a way to just check what the RNA quality of your block is before you submit for actual for Visium HD processing. And so how that works is we take a 30 micron section of your block, you submit that to us, or you can do it yourself. You do an RNA extraction, and then you run that on the bioanalyzer or tape station to look at the trace. If you have extra sections, you can do tissue morphology assessments. So you can do a DAPI stain and an H&E stain. However, if you don't have any extra sections, they recommend that you just go for the RNA quality control using the RNA extraction method. So the way we assess this trace is by looking at either the RIN, which is the RNA integrity number for fresh frozen samples. So for fresh frozen samples, because they're not fixed, they have higher quality RNA and they have intact 28S and 18S ribosomal peaks. So the RIN number looks at a ratio of that, and we look at that number. However, for fixed frozen and FFPE samples, the RNA is very degraded due to the fixation process. And so there aren't intact 28S or 18S peaks. And so we use another metric called the DV200. So what the DV200 is the percentage of fragments that are greater than 200 base pairs. So for FFPE samples, we're looking for a DV200 of minimum of 30%. The red line indicates where the 200 base pair would be, and it would be measuring the area of the curve that is to the right of that. So we want a DV200 for FFP samples that's at least 30% to continue with Visium HD. For fixed frozen samples, we're looking for a DV200 of greater than 50%, and this is all based off of validation that 10x did before creating their protocol. And for fresh frozen, we're looking for an a RIN number of greater than 4 at least. However, it's still like up to your discretion if these are precious samples and you still want to continue and see what your data looks like. Some people still do. If you have extra tissue sections, you can look, you can do a DAPI stain to just assess the quality. A stronger DAPI signal would be indicative of higher quality to have an intact nucleus and just better quality cell. And then a weaker DAPI signal would be an indication that it's not as high quality. So just a little bit about tissue preparation. It's really important that you follow the exact guidelines that 10x recommends in terms of tissue preparation as they validate it to make sure that your slides stay on your sections stay on the actual slides during the Visium HD processing. It's a very intensive process with a lot of washes and a lot of handling. So if the process isn't followed exactly as recommended, a lot of the times what happens is that tissue sections fall off during processing, which isn't ideal. The recommended section thickness for FFPE samples is 5 microns, and for fixed frozen, it's 10 microns, and for fresh frozen, it's 10 microns. However, they do have some slight guidelines depending on the type of tissue, if you should go for a slightly thicker or a slightly thinner section. So all that information is found on the 10x website, and you can ask me after if you'd like. So once we have the actual sections, we're ready to get to processing. So for FFPE samples, we have to go through deparaffинization and rehydration steps before we start H&E staining. For fixed frozen, we have to do rehydration steps. And then for fresh frozen, we have to do a quick fixation. And then we're ready to H&E stain. We can also do an IF stain. I'm just referring to H&E staining as that's more popular of the two. So once we get the H&E stain, if you have a serial section, that's ideal because then you can assess the tissue morphology before we do the H&E staining, or you can consult with a pathologist to see if the tissue section looks the way you would like it to look. For us as research technicians, we're just looking to see if there's any cracks in the tissues, any folding, because you won't get any data from those areas, as well as any lifting even if the lifting is outside of your region of interest, during the processing, if that tissue lifts and folds over onto your region of interest, it's not something we can really control. So we'll let you know those risks before we start processing. 10x also recommends that you look for things like hemorrhages, cracks, oxidized blood, tissue necrosis, things like delayed fixation, so the time post-collection of the sample to when it's put in fixative, how long you fix the samples for, these can all affect the quality of the sample that you get. So these are things you can look for before you start using the expensive reagents, which happens after this point. So now we're ready to go into decross-linking and prope-hybridization. So at this point, we put the sections in a cassette, and then we have it over your region of interest, and that's how we're adding and removing reagents. Before we do decross-linking, we quickly remove the H&E stain and the staining process. And then for samples, only FFPE samples and fixed frozen samples undergo decross-linking. And that's because the fixation process, putting them in formalin or paraformaldehyde, this cross-links the RNA. And so to access that cross-linked RNA, we have to go through decross-linking. So this is at 80 degrees for 30 minutes. We skipped a step for fresh frozen samples. After that, we're ready to get to probe hybridization. So as mentioned, HD is a whole transcriptome gene expression, allows for whole transcriptome gene expression, which means that there's around 3 probes per gene. And there's more depending on, there's slightly more for some probes and slightly less depending on the isoforms and the expression levels. So we add, depending on your sample type, human or mouse probes, there's left-handed side probes and right-handed side probes, and they both need to bind to the mRNA target site. The right-handed side probes have a poly A tail. And then overnight, we do probe hybridization because it needs to sit on the thermocycler for at least 16 to 24 hours at 50 degrees. At this point, we're ready for ligation. It's the next day. We add a ligation enzyme and a ligation buffer, and this allows the probes to be ligated together and form a complete sequence. Then we're ready for the transcriptomic probe transfer, and this happens in the actual site assist. There's a couple washes, like we do some post-hybridization washes, some post-ligation washes before this point, and we also, so with V2 version of Visium. The slides were kept at room temperature, so they were ready to load into the cytosist whenever. However, the HD slides are kept at -80, so we need to take them out, thaw them, wash them, and equilibrate them to room temperature before we start using them. And then I have a video, I'm not sure if it's gonna work. Oh, it's not here. I had a video that was gonna show me loading the site. Oh, is it playing? Oh, it's not showing here. Let me just start it again, because now I'm not sure where. That shows me actually loading the cytosist. So on the bottom horizontally is the Visium HD slide. It's drying. At this time, I'm putting the sections on. So A1 is on the right, D1 would be on the left, and I'm aligning it. It looks really close, like I'm touching, but I'm not touching the tissue section. I'm just aligning it to make sure that the region of interest is within a black square outline that's there. And just to make sure that the ROI is captured. After the Visium HD slide is fully dry, I make a probe release mixture, and that has an RNase treatment and a permeabilization enzyme. And once you make that mixture, you have to load the cytosist within 5 minutes. So I add that solution, which I'll get to now pretty soon, but I'll add that solution in the 2 spacers that are on the Visium HD slide at this point. And then I will close the machine and start it, and it'll be at 37 for about 37 degrees, sorry, for about 30 minutes running the cytosist capture. So on the left, that diagram shows what's happening in the cytosist. So horizontally, you have the Visium HD slide that has the 2x2 micron bins, and then you have your section with the ligated probes that's directly on top of the capture areas. And so what happens is that probe release solution with the RNase treatment removes the ligated probes, and then they bind to the oligonucleotide sequences that are on the bins on the HD slide. And so it's kind of hard to see, it's a bit small. On the slide, you have the oligonucleotide sequence that has the UMI, which is a unique molecular identifier, the spatial barcode, and a polydetail that binds to the polydetail that was on the right-handed side probe. And this is what happens during the cytosist capture. So once the cytosist is done, it produces a lower quality image. We look at this image to just see if we aligned correctly, and then later use this image to align onto your higher quality H&E image. The outside is the fiducial frame, and we use that for alignment later. So now we're ready for probe extension and library construction, and then moving on to sequencing. So extension happens on the thermocycler for an hour. We add extension enzyme and extension buffer, and that extends the ligated product with the oligonucleotide sequence from the actual Visium HD slide. After that, we have provolution. So we're removing all the products from the slide onto and moving them into a PCR tube. So we do this with a 0.08 molar potassium hydroxide solution. We incubate it with that, and then we remove all of the products from the slide into an actual PCR tube. After that, we have preamplification. So this is just using PCR to amplify the product, and a SCRI select cleanup. SCRI beads are solid phase reversible immobilization beads. They're used for purifying nucleic acids. They have a hydrophilic coating, which allows for reversible binding to nucleic acids, and depending on the ratio of the beads to the sample, that will determine the size of fragment that we get. So it's used for size selection as well, which is great. So that allows us to remove impurities and select for the size of the preamp product that we want. So once we have a preamp product, we can store it at -20 for 4 weeks, maximum 4 weeks before we make the library. So usually we'll pull a bunch of samples and make a bunch of libraries at the same time. So now we're ready for probe-based library construction. At this point, we use qPCR of the preamp cycle to determine the CQ value, and then use that to determine the cycle number that we're going to use for the library PCR. So for example, if we get a CQ value of 8.2, 10x has determined that you do the CQ value rounded up +1, and that's the cycle number we use. So it would be 10 cycles for the library PCR. And then we would run that library PCR. So in that library PCR, we would have the master mix. We'd also have the indexes that we're going to use for sequencing. So it's dual indexed. So I5, I7 indexes would be added. And then we'd also have the preamp product. After that's all run, we do another cleanup with SCRI beads, and we have our final library, which has the dual index insert, as well as our probes, as well as the spatial barcode and the unique molecular identifier. And at this point, it's ready for sequencing. So before sequencing, we do a quick QC step of running it on the bioanalyzer just to see the trace. We're expecting our trace to be between 200 and 300 base pair. This is really sharp peak. This is like a perfect trace. If we had any sort of over amplification or beads still in the product, we'd be able to see it at this point. And then we have extra preamp product that we can go back to and remake the library. And after that, it's ready to go for sequencing. So we use Illumina sequencing, usually the Nova X because we have the high throughput as a genomic center. We do, which is short read, next generation sequencing, paired end sequencing with the read 1T and read 2T, and then you would get your, and that's when we get the web summary data. So in terms of sequencing depth, 10x recommends 275 million reads per fully covered capture area for FFPE samples. We find that's really conservative for most samples, so we aim for 500 million reads. For fresh frozen, they recommend 700 million reads per capture area, fully covered capture area. And for fixed frozen, they recommend, I think, 500 million reads for fully covered capture area. And then depending on if you're not using the whole capture area, you can do less or more. But yeah, we aim for a little bit higher. We find that it works for more tissue types. So at this point, we're ready to we get the web summary and look at some key metrics before we send you the data. So the first thing we look at is the mean UMIs per 8 micron bin. So usually we see a lot higher mean UMIs per bin in fresh frozen samples versus FFPE, but ideally, like depending on tissue type, we want at least 100 mean UMIs per 8 micron bin. That if it's lower than that, then it raises a little bit of a red flag and look more into the data to see what happened. However, tissue and sample quality really determines what your mean UMIs per bin will be. So sometimes people let us know, like, we expect this, the UMIs were going to be really low for these samples. Another thing we look at are the reads mapped to the probe set, as well as the reads mapped confidently to the probe set. So the reads mapped to the probe set are the percentage of reads mapped with a map Q that are greater than 0. And then the confidently mapped to the probe set are map Q equal to 255, which just means that it's a single unique probe set it's bound to a single unique probe set. If we see, so ideally over 80% would be okay. If we see slightly lower, it could be indicative that something happened with heated washes, or it could also be sample related. So it's something that we just look for. Another thing we look at are the reads half mapped to probe set and then the reads split mapped to the probe set. So we want those to be low because they're not fully, the left-handed side and the right-handed side are not fully bound to the probe sets. Another thing we also look at are the number of reads. We're aiming for a total number of reads that gives you a decent saturation. So if we were aiming for 500 million reads, we saw that it was a lot less, then we would, depending on the sequencing saturation, top up the samples. Sequencing saturation just determines how much of that sample, sequencing saturation is dependent on your library complexity and the percent of usable UMIs that you have. So that would determine saturation. And then you can also look at the sequencing actual curve. And that can determine whether it's worth doing more top-up sequence. If it's flattened at the top of the curve, then it's not really, you might not get a ton of more data by top-up sequencing. However, if it's more linear, then you can probably benefit from some more sequencing. We also check here to see if your tissue is detected and that there's proper fiducial alignment. So sometimes for most samples, we manually align them. And so we just verify that the red circles are all around the actual fiducial frame. And this will just make sure that your alignment is correct with your data. You can also look at the clustering of your data at the 8 micron and 16 micron resolution. And this will just give you a better idea of if the sample is how you expect it to look at this point. So yeah, this is my presentation on the processing portion of Visium HD. I hope you guys understood better, like have a better understanding of the wet lab process. Thank you for listening. And this is our contact information. Farzaneh is also someone who works on our team for spatial transcriptomics. She also does Xenium. And then Troy, who's the head of operations of the Princess Margaret Genomics Center. So feel free to reach out if you guys have any questions.